Thank you all for the opportunity of sharing with you some of the work we are doing here in Brazil. I will talk about why we should employ X-ray spectrometry in plant science and present some of the challenges in tracing elements and making chemical speciation. More specifically, I will discuss why we should study plants, try to describe how X-ray fluorescence spectrometry and X-ray absorption spectroscopy can be useful tools for plant science, and try to deep a little bit in two topics, which is sample preparation, which is something challenging while preparing samples for X-ray spectrometry, preparing biological samples like plant samples for X-ray spectrometry, and try to show examples of studies in dynamic conditions. That said, I'm at the University of São Paulo, more specifically in the countryside of the state, in a city called Piracicaba, which you can see here. This is 400,000 people town. It's a medium size in Brazil. We are surrounded by the sugarcane industry. It's something interesting because in Brazil, when you go to the gas station with your car, you can choose between using ethanol or gasoline. This is what we call a choice in the pump. And an important part of the ethanol produced in Brazil comes from this area. And specifically, I'm at the Center for Nuclear Energy in Agriculture, which is attached to the College of Agriculture of the University of São Paulo, and was created 55 years ago by institute, by some visionaries that wanted to use radioisotopes, X-rays, and stable isotopes to investigate plants and soil. And what exactly we do here? We try to understand plants and try to make them work better, which is an ambitious goal, yeah, because we believe that plants and agriculture have a source of wealth, and if this is properly managed, we think that it can contribute to create a sustainable wealth, which is something very important. Our research group has basic three axes of action. We employ X-ray spectrometry to perform soil and plant diagnosis. It means we are interested in investigating or measuring the nutritional status of soils and plants, and then one could rationally decide whether fertilizer should be added or not. We investigate mechanisms of uptake and transport of mineral nutrients in plants. And another branch which deals with nanotechnology in agriculture, which consists basically in investigating what nanoparticles can be absorbed, transported, and used by plants. And we do it like Professor Dean Hesterberg used to say. He used to say across length scales. We employ X-rays to understand fundamental phenomenon. When it works, we go in scale up in field and try to apply. Like, for example, there are two small companies issued from this lab founded by former students. And why X-ray spectrometry could be useful for agriculture? I will give you some examples. For example, in seed treatment, this is a very common practice. Before sowing, seeds can be treated with micronutrients. Here you can see soybean seeds, and maize seeds. And those seeds here on these bags were treated with copper, zinc, and molybdenum, which is intended to increase germination rate or enhance germination performance, yielding more productive plants. And it's not still clear of how much of this fertilizer applied to the seeds penetrate within the seed and is going to be used during the germination process, or how much of the fertilizer will stay in soil. So we can use X-ray spectrometry to map it and look at what these nutrients will be attached outside, or they are going to be immediately metabolized and employed by the seed. Here we see another example, because we could use X-ray spectrometry to investigate the dynamics of fertilizer in soil. You see this very small pellet here. This is this bead. This is a pellet, a granule of phosphate fertilizer. And we can use spectrometry to trace how fast this phosphate fertilizer is going to leave the granule and reach the roots. And tropical soils, usually they have a feature which is very different from soils from the Northern Hemisphere, because they attach the phosphate, and phosphate will bind to soils and become not available to plants. Here, and in the Northern Hemisphere, is the opposite. The soils will not bind to the phosphate, and it ends leaching and causing environmental problems. So this here is an image recorded at the Brazilian synchrotron, showing the phosphorus distribution in the granule and in the nearby soil. We can also use X-ray spectrometry to investigate the uptake of nutrients by root and this movement from root to shoot. So we can measure it. And for example, if we put here zinc sulfate, in which chemical form, because zinc is a chemical, is a nutrient for plants, in which chemical form it's going to leave the soil, penetrate the roots, and move inside of the plant. And what if one employs a zinc nitrate, for instance? Would that be different in terms of efficiency? And this efficiency can be explained by chemical species. Additionally, when plants, they are going to produce fruits, or when they are going to die, nutrients, some of them, they would leave certain regions of the plant, and this is called the nutrient remobilization, and go to other parts of plants. And what are the phenomena that triggers this movement? And can we use X-rays to investigate it? Foliar fertilization is another example, because we apply fertilizer on the leaf surface, and this fertilizer has to cross cuticle, epidermis, enter inside of the leaf, and be used by the plants. So those are examples of phenomena that can be explored using X-ray spectrometry. And last but not least,
Hudson, sorry to interrupt. You have a drawing still on your screen. Maybe you need to go into annotate and hit clear.
Okay. Yeah. There is the drawing. Yeah, but I don't know how to remove it.
So if you go to the top of your screen, there might be something that says view options, and then annotate.
View options, annotate.
And then you'll see a little garbage pail to clear all.
Drawings. Yeah, but I was using the PowerPoint. No problem.
Well, maybe just reproject now and it should be gone. Yeah. I apologize. Yeah.
No problem.
Good. Yeah.
So I was talking about biofortification, yeah, because we can. This is, for example, this is a rice grain, and here this is the embryo and the endosperm. And usually we eat the endosperm, because when rice, someone many years ago had the idea of polishing the rice, And this aileron layer, the bran, is removed, and the embryo is removed as well. And we eat this part here. And some of my colleagues, they mapped the spatial distribution of selenium in biofortified rice grains. And we see here the spatial distribution of selenium. So those are examples of where X-ray spectrometry could be applied in agriculture. And let me just erase it here. And then one could perhaps wonder, but we already know enough. Yeah, we know how plants grow. We know that they need light, carbon dioxide, oxygen, water. And here I share with you some data showing the productivity in tons per hectare. And here we have time. Those are years. And the productivity of three very important cereal crops: wheat, barley, and oats in the United Kingdom. So we can see here from the 13th century up to nowadays, and we see that productivity was stagnated until almost the 17th century. Less than 1 ton per hectare. So, and then it started to increase. It may be, one could think it's a coincidence, but it's more or less the same time of these movements of enlightening and science, flourishing of science. And then productivity starts to increase. And let me just take a pointer here. Yeah. So productivity starts to increase more or less here. And then in this period here, which is highlighted by this square, lived someone that we know as one of the first economists, Thomas Malthus in the UK. And he was very worried because he noted that productivity was increasing, but he realized as well that the population was increasing much faster than our ability of producing food, of growing food. And he was worried about a kind of a catastrophe that would shortage of food. But he could not foresee that here in the 20th century, Fritz Haber and Carl Bosch would develop a method of extracting nitrogen from the atmosphere and transforming it in fertilizer. And turns out that nitrogen was one of the most limiting factors preventing plants to preventing plant yield or limiting plant yield. And other things happened during the last century, like the mechanization, pesticides, genetics, and all that contributed to a huge increase in productivity. So we live here at the beginning of the 20th century, producing more or less 2 tons per hectare, and we multiplied that by 4 or by 2. And what's curious, if we look at the past 20 years, productivity of those crops seem to be stagnated again. And you know that world population is still growing, and this population, this growth, is happening especially in developing countries. It means that those countries are developing, and those people will want to consume more, which might put pressure on the production of food. But more than that, agriculture is being more and more responsible for producing fuel, fiber, raw molecules or platform molecules for chemical industry, and all that pressures agriculture for increasing outputs. Some people estimate that by 2050, outputs should double. One could say, okay, so we can extend the planted area. The problem is that there is no more room for extending planting area. Otherwise, one would have to cut forests or native biomes, which is not a good idea. So we have to make plants to be more productive. It means to produce more, use the same unit of area. And I believe that if we understand better how plants work, how they function, we can make them to be more productive in a sustainable way. And I will give you an example, which is a study that has been performed in partnership with our colleagues in Spain. And it's being carried out by one of my PhD students. Our colleagues in Spain, they developed hydroxyapatite nanoparticles. And when they cover those nanoparticles with urea, which is a source of nitrogen, is a fertilizer, it increases a lot the use efficiency of nitrogen. And then we decided to understand how it was happening. And they prepared the sum of those apatite nanoparticles, replacing part of the calcium by strontium, and still covering it with urea. And strontium in this case is a tracer. And then we applied a droplet of the fertilizer here, of this nanoparticle containing the urea on the top of a leaf surface. We are using wheat as a model plant because nitrogen is an important limiting factor for this crop. Like you can see here, a sample holder. And then you see here a droplet of the suspension, the colloidal dispersion of the nanoparticle containing the urea. And then we use a benchtop machine, an X-ray fluorescence spectrometer, to trace whether these nanoparticles were being absorbed. And then by performing line scans here, we could measure in green, this is the absorption of the nanoparticle labeled with the strontium, and here a positive control, which is the strontium chloride, which penetrates much faster within the leaf. But now the question is, how could one check whether the nanoparticle was absorbed in its pristine form? I mean, the nanoparticle, perhaps it was dissolved. And just by using X-ray fluorescence spectrometry, we cannot tell anything about the chemical species, how the nanoparticle is being absorbed. For that, we need X-ray absorption spectrometry. But this is a clear example of how X-ray spectrometry can be used for in vivo studies. The other alternative, perhaps, could be using radioisotopes. But it's very difficult working with radioisotopes. One could prepare this nanoparticle using the 32 isotope of phosphorus, which is radioactive, but it's very difficult, manipulate and dangerous. X-rays is a much safer way of doing it, and versatile, because while synthesizing the nanoparticle, one does not have to manipulate those radioisotopes. And then the question is, this study was performed in vivo, yeah? But should we do ex situ X-ray spectrometry or in vivo one? And both, of course, have advantages and disadvantages and limitations. As an analogy, for example, we learned a lot from ex situ studies. this is an example of an ex situ study. Yeah, there is a corpse here, and people studying this corpse. I would say that this is an example. This is a nodule produced by a soybean plant, which is something amazing, because soybean plants and all other legumes, they do not require nitrogen fertilizer. Inside of the roots of those plants, those roots can be infected by certain types of bacteria, good bacteria, which will fix the nitrogen from the air and do what the Haber-Bosch process does in an industry inside of the root of the plant. So the bacteria, they require sugar in exchange. They require carbohydrates in exchange, and they give chemical available forms or reactive forms of nitrogen to the plant. And this is a cut of this nodule. And we see here the spatial distribution of sulfur, magnesium, and chlorine in this nodule, this factory. This is a biofactory of nitrogen, of a nitrogen fertilizer. And this is an example of in vivo study. Yeah, someone alive in a tomography, and just like this rat. In this study, they injected molybdenum oxide nanoparticles inside of the rat, and they did X-ray fluorescence tomography. And we can see here the same rat, 15 minutes after injection, 30 minutes, up to 14 weeks. And there is still some molybdenum in the liver of the rat. And when I show this example to the students here, and I say, take care of your liver, yeah, do not drink too much. Because you see, everything went to the liver of the rat, of the mouse. And so this is an example of in vivo. So we can learn from both procedures, but in vivo has some striking advantages, because I can follow the same organism. I can monitor the same organism. And there is a huge variability between individuals of the same species. I can take soybean plants and use 5 plants, and I will get 5 slightly different responses from the plants. Luckily, slight different responses, because they are different individuals. But using or studying the same individual is a striking advantage. And we can trace dynamic process. We can see it while it is happening. And material scientists, they realized it many years ago. Everyone working in catalysis, I worked in catalysis from 2012 up to 2014 at a group of Professor Grunwald in Germany. And he was doing catalysis in situ and under operando conditions already for many years, because material science realized that it's much better monitoring or following a process while it's happening. So we learn more in this situation. So it's much easier saying than doing, but we working now on biological material, we have to make same efforts to move towards in vivo X-ray spectrometry. But as I said, it's challenging. So we should have a break now.
Yeah, let's. So I'll start with a few questions while people are typing their questions in. First, obviously what we're talking about here is often element-specific tracking, trying to figure out where nutrients go or where toxins go or what have you. What are the relevant concentrations for the species that you're interested in?
Yeah, for example, if you are talking about macronutrients, which would be phosphorus, calcium, potassium, we are talking about thousands of milligrams per kilogram. Mm-hmm. In average, this is an important detail. And for micronutrients such as zinc, manganese, iron, we may talk about tens of milligram per kilogram up to few hundreds of milligram per kilogram. But the spatial distribution of those elements, they are not homogeneous. So depending on the structure, like a seed, we will see that in certain locations, concentration of potassium can reach tens of thousands of milligram per kilogram. As well, the concentration of manganese or zinc can reach few thousands of milligram per kilogram.
I see. Okay. So some things are easy, some things are hard. Seems to be the important detail, and no doubt the spatial distribution tells you something about function and and life cycle of the plant and whatnot. Okay. Gabriel, you have a question. Can you unmute your mic?
Yes. I'm wondering about the main operational challenges for carrying out in vivo XRF analysis of plant tissues. Which are the biggest challenges, Professor Hudson?
Yeah, we'll show some of them. Yeah, this is a very good question. We'll discuss some of them later, but I can put in advance that we should not burn the sample, under high flux of X-rays, because the amount of energy deposited on the sample can just burn it. But I would say that's not a challenge different from any other scientist, which is the compromise between modifying what you are studying while you are studying. But biological materials, they usually are more sensitive than soil, for example, or than minerals, or than catalysts. I remember that working with catalysts, depending on the beam line, we could clearly see the reaction rates increasing when X-ray beam was on, just because temperature was increasing. So this is the challenge: how to investigate the sample without modifying it. And this is tricky, but I will show some examples later, talking about radiation damage.
Great, thank you.
All right, very good. Professor, you should continue.
Okay. And then let's consider the case in which we are doing ex situ studies, which is, I would say, is still challenging, but we have better control of radiation damage. So how to prepare and how to preserve the samples for ex situ studies. So suppose one has a leaf sample, so plant leaf, and then we have to cut it, and differently from people working on traditional anatomy, we have to freeze the sample. We have to just try to keep everything like it was. But this freezing has to be as quick as possible. Otherwise, the water inside of the sample will form crystals. Anyway, it will form crystals. The water, but you wanted to get the crystals as small as possible. Those ice crystals, because if the crystals are large, they are going to destroy the cells and membranes, and then what we are going to measure does not correspond to the reality. For that, we have to use a non-volatile liquid. So just plunging the sample in liquid nitrogen might not be a good idea, because nitrogen is highly volatile and the sample is warm, although nitrogen will be at minus 180 degrees Celsius, but it is highly volatile. And then the heat transfer from the sample to the nitrogen will be too small or too low. And one is going to probably get large crystals inside of the sample. And try to use a small piece of sample to freeze it. So this is the first step. We have to quickly stop everything. And it's good to have someone like people from anatomy, plant anatomy with you, but keep in mind that those methods used to prepare a sample for classical anatomy, which aim at preserving the structure, they are not necessarily suitable, or they are most likely not suitable for X-ray spectrometry, because they are going to infiltrate the samples with liquids, and then everything which was inside of the sample is going to change place. And then an XRF map or an XAS spectrum may not reflect the reality. So then once the sample is frozen, one has to embed it in a resin and cut it, which is also challenging, because depending on the X-ray beam size, for example, typically in those fourth generation synchrotron machines, one has a beam size of 1 micrometer, 2 micrometer, or even smaller. The samples cannot be thick, otherwise the lateral resolution doesn't make sense. So you have to cut the sample using a cryostat, and already in the moment of putting the sample in the resin block, we have to put in the right position. Do not allow the sample to melt, otherwise it's going to freeze again slowly. And you can check the quality of the cuts using the microscope. And then once it is cut, you have to preserve it at a low temperature, go to the beam line, and make sure that they have a cryostat there, or a cryojet, for keeping the sample cool while measuring. And every manipulation step has to be quick. You have to be as quick as possible. So this is a big challenge. I do recommend anyone who wants to try or start doing it to read this review by Hiran Castillo Michel. He's a beamline scientist at the ESRF. It's a very good review of telling the pros and cons of each method. Then I will show you an example of a frozen hydrated sample, which is this sample that was, as the name say, it was frozen, but it's still hydrated. This was done at the LNLS, at the old QUVX machine. Was a master thesis of one of my students, and she was investigating the uptake, root to shoot uptake of zinc in plants, zinc nanoparticles and zinc sulfate, and using X-ray fluorescence in our lab, she could monitor the increase in zinc concentration in leaves as a function of time. So for nanoparticles of different sizes and using also salts. And once we assured that we had zinc in the leaves, like we see here in this image, we went to the synchrotron. And the sample was prepared like I told you, and we can see here a leaf, but we did not cut it because the X-ray beam size we had at the Brazilian synchrotron at that time was large. You see here, 450 by 250 micrometers. It was not a beam line dedicated to microscopy at Xav's tubing line. And this is the cryojet, keeping the temperature low. And then we can see here that regardless the treatment applied, either we gave to the plants zinc sulfate or zinc oxides nanoparticle, the chemical species in the leaves were pretty the same. It was a mixture, it could be fitted, yeah, something like or similar to zinc bound to histidine, which is an amino acid, and zinc phosphate. So this is an example of a frozen hydrated sample. And then challenge number 2 is moving to dynamic systems, yeah. And it's funny because up to 2015, I moved here in 2015. R&D used it to be research and development. And then since I started studying plants, it became radiation and damage, which is something that we have to be very careful of. And if you have any questions, Professor Seidler, or or we could move forward and
I think let's move forward. This is very interesting. Yeah.
Perfect. So this is an example. This is a study that we have been doing together with Professor Michael Jones in Queensland University of Technology in Australia. Because they have also there is a very productive group there studying plants, and they employ X-ray spectrometry for studying plants, and they face kind of same challenge, the effects of X-rays on a living leaf. And we are doing some studies here using, for example, a polychromatic X-ray tube. So this is a tube that works from up to 50 kilovolts and 1000 microamperes. And here in this condition, it was working at 40 kilovolts and 950 microamperes. And what we see here is the effects, or you see this circle here. This is where the X-ray beam is. It was a 1 millimeter X-ray being unfocused directly from the source, so almost 41 watts of power. And we see here the signal of phosphorus, potassium, and calcium. And each of those points were either 60 seconds or 120 seconds of dwell time. And the total exposure time was 60 minutes. And we do not see any clear change in the chemical composition of the irradiated spot. But when we take this same X-ray tube and we put a polycapillary in front of it, having a 30 micrometer focused X-ray beam, which would give a flux density nearly 1,000 times higher than in this situation here, we clearly see a decrease in the signal of potassium and an increase in the signal of calcium, increase in the signal of phosphorus, and a decrease in Compton scattering. So clearly, the X-ray is damaging the tissue. So this is one of the I would say, if you could call symptoms or signs of radiation damage. The composition of the tissue is going to change. Remembering that this leaf is alive. Yeah, it's a living plant. And then what we did next was to put primary filter in front of the X-ray tube, a titanium X-ray filter, which would increase our sensibility or sensitivity for micronutrients like manganese. And then what we see is that in addition to what we saw before, which was decreasing of potassium, decreasing of Compton scattering, which might indicate dehydration, water is also living. In addition of calcium increase, we see also manganese increase in this area. Yeah. So manganese also increased under irradiation. 100-second shots, but and here a total exposure time of 60 seconds, and we do not see the same trend. So it's a matter of dose. So what matters is radiation dose. So at lower doses, we do not see measurable damages, but under high dose, yes. And unfortunately, I cannot tell you what is the photon flux density here, because I do not have a diode for measuring, but then we report what was the power used. Then we can compare between exposure time and the power. And then what we did is what we decided to map the region which was just irradiated, and we irradiated for 20 minutes on a spot using this focused X-ray beam, and then we mapped very quickly is still the fresh tissue. So this is the image we get for potassium. So we see a hole here, yeah? Potassium really moved around, so we left the irradiated area. While it looks like that a spot of calcium was formed here and a large spot of manganese. And this leaf here below, it was irradiated only for 10 seconds, 1 point, and then we mapped again the tissue, and the dwell time during the mapping was like 2 seconds per point or 1 second per point, very quickly. And then we do not notice the same damages. Then one could say that this hole here, this absence of potassium, or this accumulation of manganese was caused because this area here dried, yeah? One could say, so the water left, then the concentration of certain elements increased. Then what we did, we got this same leaf and dried it. We put it in an oven for, you see here, this the region, the damaged region. We put it in an oven for 24 hours. And we dried the leaf, removing the water, and you see it's more or less the same region, the same area. This vein here is this one here. And now we see a slightly different image for potassium, because while drying, the liquids are moving and some nutrients are highly mobile. So drying the sample in an oven is not a good preparation method for X-ray spectrometry, yeah, because we're going to modify it. But the question here was, this manganese spot was caused because the tissue was dried, and we see that the background here of manganese increased in the dried leaf, but the spot still remains, which suggests that manganese was going to the region which was being irradiated. By the way, this is a soybean leaf. Perhaps I did not say it. So it looks like that some elements go away and some elements go in. I do not have enough time to show, but we did also some histochemical tests, and it's clear that a lot of enzymes related to the antioxidant metabolism were active here, and manganese is a cofactor of some of those enzymes. This is an example of a radiation damage study done at the Brazilian synchrotron. I just lost my mouse here. Okay. And we are looking at the Compton scattering, just looking at the vein and in the blade, irradiating for a couple, we're recording Zane's spectrum, nearly 20 minutes per spectrum, and we do not see any change in the Compton scattering while measuring the spectrum. So which suggests that the sample was not dehydrating, but pay attention to the X-ray flux here, 10 to the 8th photons per second per square millimeter. So it was monochromatic radiation. And then we did another study at the Brazilian synchrotron, now at the XAFS2 beam line, using a larger beam, 6 minutes per spectrum. And what we noticed, you can see here what we called point A. It means we measured one spectrum in point A, then one spectrum in point B, and then a couple of spectra in point C. And then we clearly see that the spectrum in point C starts to change after nearly 12 to 18 minutes of irradiation. So a spectrum changing means that the chemical neighborhood of the target elements is being modified. So we see radiation damage after 12 to 18 minutes of irradiation. The X-ray photon flux was 10 to the 9th photons per second per square millimeter, so the density flux. So this is a study published by Professor Jones of Queensland University of Technology. And then we see here he was increasing the dose. So we see, let's look at potassium. Potassium So this area was not mapped, and the area below was mapped both areas were mapped, sorry. But here he increased the dose, yeah. So we see here the radiation dose given in gray increasing, and we just see that potassium is moving away. He employed sunflowers, but we do not see increasing of calcium. But potassium is moving away. But look to this is the X-ray beam, 1 to 5 micrometers, and this is the flux density, 10 to the 10th photons per second per square millimeter, per square micrometer. When we convert it per square millimeter, you see the difference of flux density compared to what we have in the Brazilian QUVX machine. So it's a 7 orders of magnitude higher. It means that we should not forget that those under the same photon flux density and at the same energy and for the same sample volume, the dose will be directly proportional to the time of exposure. And it means that with 1 second exposure in Brazil, in the old Brazilian synchrotron, we would have to, 1 second in Australia would be equivalent to millions of seconds of time of exposure in the old machine. So this is why it was suitable for in vivo study. But now in this new machine, the Sirius one, which is going to give a photon flux of this order of magnitude, I'm afraid that it's not going to be possible of performing in vivo studies anymore. So an example of in vivo XAS done in the Brazilian old QUVX machine. You can see here, for example, the student just spreading the fertilizer in the leaf plate, and he would monitor the chemical species of the fertilizer in the petiole, which is the part of the leaf that, yeah, the petiole. I would not. It's kind of. I do not know another word in English for it. But then he would do the chemical speciation here. Would apply different chemical species of fertilizers like sulfates and chelates and see how they are transported. And it was possible to do it in vivo. And Bianca, for example, she did her master's here, and she was interested in investigating the relationship between manganese and glyphosate. Because glyphosate is a herbicide and is by far the most successful herbicide ever. But somehow it seems that it prevents plants of absorbing manganese. And what she was doing was spreading the manganese fertilizer here in the leaf. And we can see an X-ray spectrum before the application and 12 hours after the leaf received the fertilizer. So the concentration of manganese here in the petiole increased. And then she measured it here, and we see here time and the number of counts of manganese normalized by the Compton scattering, the Thomson scattering, just to be sure that we would not get any artifact of sample thickness, because the position of the measurement could be slightly different. And we clearly see that depending on the source, like phosphate, sulfate, the uptake of those fertilizers were reduced by the glyphosate. While when manganese is complexed by or chelated by EDTA, it's not affected by glyphosate. And then she did a similar thing. She spread the fertilizer here in the leaf, and then we went to the XRF beam line, and she got a couple of spectra in different points, 1 spectrum per point, neighbor points, of course, to avoid those radiation damage. And then what we see, so this is a typical experiment, X-rays This is the area measured. X-ray would come from here. This is a X-ray fluorescence detector. And this is it. So when the plant receives manganese EDTA inside of the plant, inside of the petiole, the chemical species of manganese looks very much like steel manganese EDTA. While when we apply phosphide and sulfate, we found a more similar chemical species, which was closer to manganese bound to organic acids. And what was curious is that we do not see any signs of glyphosate binding manganese inside of the plant. So it's clear that glyphosate prevents the uptake of manganese, but what looks like is that they react outside of the plant and forming a non-soluble compound, which is not going to be absorbed by the plant. So my final remarks. So I think that this is a very interesting subject and there are lots of topics to be addressed, and we need people working on that for the reasons I told you at the beginning of the presentation. We need to increase productivity using, but not by increasing the use of inputs like fertilizers. We have to increase the unit of biomass and grains per unit of land and unit of water and unit of fertilizers. Sample preparation for high flux density of those microbeans, high flux density microbeans, is not a straightforward task, and is not just taking the procedure with very consolidated procedures for anatomy. Otherwise, we are going to look at artifacts. We should be very careful with radiation damage. Again, for not being fooled by artifacts, and then this issue of in vivo measurements. So I have the feeling that scientists are pushing for brighter synchrotrons, brighter X-ray sources, but I not feel the same movement towards more efficient detectors. I think that we need faster detectors because of those high X-ray doses. We need better detectors, larger areas, faster, much better detectors than the detectors that are currently available. Otherwise, we are building very powerful synchrotrons that will burn our samples faster, and then we have to struggle against burning the samples, or just put a filter in front of the X-ray beam to reduce the flux density. It's just a nonsense. So the development of detectors have to come at the same pace as the brightness of synchrotrons. So I finish here. I thank you very much. The funding agencies that fund our studies, the companies that fund our studies, and to the students and colleagues that are collaborating, and without them, I would not be here showing those results for you. Thank you very much.
Very interesting, thank you. To start the questions, Matthew, you had a question?
Yes, in the early days of work with microprobes and plants, people used to do freeze drying all the time. And I was wondering just how bad that is. Does it even preserve distributions? And does it have any hope of preserving chemistry?
Yeah, good question. Because freeze drying for animal tissue is still common. For chemical speciation purposes, the biologists say that the nature of the compounds are more stable than those found in plants. But for mapping purposes, I would say that the smaller the beam, the more difficult is the sample preparation, because then any artifacts or redistribution of elements would become more evident. For example, doing spatial distribution with a larger beam, like a 500 micrometer beam, I don't think that freeze drying would be a big deal. But suppose that we have a 1 micrometer X-ray beam, then any movement, any displacement caused during the freeze drying would appear. Then we would be led to a mistake. But I think that it depends on the size of the X-ray beam. Like here in the lab, we have an equipment which gives me an X-ray beam with 30 to 50 micrometers, depending on the energy, because it's a polychromatic source. And in those cases, I would say that most of the mistakes that would be caused by freeze drying, they will not appear because of my lateral resolution.
Right, if you plunge freeze, so that it should in principle immobilize everything and then freeze dry. I wonder if that would remedy that, or at least fix the distribution in place on the micron scale.
Yeah, I would say that in hundreds of micrometer, no. In tens of micrometer, perhaps, and sub-micrometer, certainly, because the plunge freezing, for example, in liquid nitrogen, would lead to the formation of those large ice crystals. And this is a problem.
Well, I meant plunge freezing in the appropriate liquid, such as ethane.
Yeah, then in this case, we would not have large crystals, but the freeze drying could lead to remobilization. There is a study done by Anthony van der End, and he has images showing the modification in shape that freeze drying can cause in the samples. But again, all depends on the lateral resolution. The higher the lateral resolution, the more critical is the sample preparation. But thanks for the question.
Yeah, and as for temperature rise, yeah, I'm a little surprised that you find that to be a problem because, for instance, on the microprobe we have here at ALS, the power is microwatts. Now, of course, if we get to ALSU on a nanoprobe, then now we're actually talking about a blowtorch.
Mm-hmm.
Yeah, insertion devices, of course, are more like milliwatts. I guess just the last question, in terms of radiation damage mechanisms, is there any possibility you have some ozone damage happening?
I do not remember of testing of ozone, but certainly we detected hydrogen peroxide.
I see, okay.
Yeah, and other organic radicals. We did a couple of tests using those histochemical colorants that would react, and then we take it to the microscope. Yeah, hydrogen peroxide certainly.
Okay.